अच्छा इसमें
हो गया
वीडियो ऑन है कि
हेलो
वीडियो ऑफ है सर स्क्रीन शेयरिंग
हेलो
बाहर जाओ।
Hello, good afternoon, dear students. There is some technical problem and we could not start in time. Hope you must be having patience to wait for such long time. Anyway, this is the COVID time and we are passing through a very second phase of severe form of COVID. So stay home and stay safe. Now, whatever teaching we can conduct through internet, we'll try to learn something. And today we are going to start a new chapter, carbohydrate metabolism. After carbohydrate chemistry, we are going for metabolism of carbohydrate. And so let us start slowly, and because of it's already 40, I was supposed to start carbohydrate metabolism, and I was planning to hello, some problem is there. The slides are not moving. Hello, Dr. Ram, please help. Slides are not moving. Okay. It is working now. It is working. It needs a. Oh, so today we were supposed to. Some problem is there. Anyway, in the carbohydrate metabolism, we'll see Dr. Ram, please come. Some problem is there. Dr. Ram, ये क्यों आ रहा है? ये ब्लॉक कर रहा है.
कैसे आया? कोई बात नहीं. ये है ना सर, ये क्वेश्चन इसको ऐसे कर दीजिए.
ये ब्लॉक कर रहा है.
हाँ नहीं कोई नहीं, वो ऐसे आप कर दिएगा ना.
अच्छा,
फिर आपको पैनल चाहिए था, ऐसे करना आपको जरूरत है.
पैनल जरूरत नहीं है. हाँ ये तो बस. ओके. अच्छा ये नीचे ये दबाना पड़ेगा जोर से.
आपको स्लाइड आगे बढ़ाना है. एक बार इस पे क्लिक कर लीजिए. अभी आप इस स्लाइड पे हैं. ठीक है.
ठीक है, okay. So yes, some initial glitches there, so we have to iron it out. In today we are supposed to cover digestion and absorption of carbohydrate, then glycolytic pathway and TCA cycle. But let us see, because already much time has been wasted. So we'll go straight to introduction to metabolism. In the enzyme chapter, we have seen how enzymes helps in conversion of substrate to a product. All the chemical reactions in the body are, most of them are enzyme catalyzed by enzymes. However, in cells, these reactions rarely occur in isolation. Rather, they are organized into a multi-step sequences called pathways. The product of one serves as a substrate for the subsequent reaction. Multiple reactions are there. So first substrate gets into product 1, that is a substrate for the second enzyme. Then product 2 is substrate for the third enzyme. Like that, it is multi-step sequence or metabolic pathways are there in the body. Different pathways can also interact, forming an integrated and purposeful network of chemical reactions. These are collectively called metabolism, which is the sum of all the chemical changes occurring in a cell, a tissue, or the body. So metabolism is the sum total of chemical changes occurring inside our body. So carbohydrate metabolism, we'll see how carbohydrate, of course, it is not isolated again. Carbohydrate is linked to protein metabolism, carbohydrate is linked to lipid metabolism also. But we have to cover chapter-wise individual carbohydrate, protein, and lipid metabolism. And last, we can integrate them, integration of metabolism. Most pathways can be classified either as catabolic or anabolic. So metabolism can be divided into catabolism or anabolism. Catabolism is a degradative process where complex polymers are being degraded finally to simpler substances, and during that process energy is being released, and so we get catabolic processes is mostly for getting energy for the body, whereas anabolic processes are synthetic processes which are being this is anabolic processes are synthetic processes where body synthesizes its own biomolecules depending on its requirement. So now coming to the next slide, pathways that generate a component is called a cycle. Pathways can be linear, but when the pathway generates a component of the initial substrate, then it's called a cycle. We'll see Krebs cycle, urea cycle, and many cycles are there where at the end the first substrate is regenerated. So that cyclic way it goes on. Man is a chemoheterotroph. That means who uses chemical reactions to obtain energy from organic matter in the environment. Man needs energy and building blocks to maintain body functions and adapt to physiological conditions like growth, pregnancy, lactation, exercise, and environmental changes of temperature, pressure, oxygen availability, etc. So knowledge of metabolism is necessary for understanding various functions of the body. Abnormalities in the metabolism, on the other hand, helps understanding the disease, which may be due to nutritional, enzyme, or hormone deficiency. So this is a brief about what is metabolism, and we'll see what are the anabolic processes. It can be divided into anabolic, catabolic, or amphibolic also, but it both anabolic and catabolic. So before that, we'll proceed to digestion of carbohydrates. Carbohydrates are consumed in diet mostly as polysaccharides. Dietary polysaccharides include starch, glycogen, cellulose, etc. Small amount of disaccharides and monosaccharides are also present in food. Disaccharides are which include sucrose. Sucrose is the common table sugar which we use daily. Lactose is the milk sugar, and monosaccharides are fructose and glucose, etc. Fructose is present in mostly fruits. So these are the disaccharides and monosaccharides present in the diet, but the main bulk is the polysaccharides, which is the starch, and cellulose is the non-digestible polysaccharide, which is mainly contributes to the dietary fiber. The intestinal mucosa can absorb only monosaccharides. So after digestion process is required because mostly polysaccharides, and intestine cannot absorb polysaccharides or disaccharides. It only can absorb monosaccharides. Larger carbohydrates are hydrolyzed into monosaccharides during digestion. So enzymes involved in digestion of carbohydrates are present in saliva, pancreatic juice, and intestinal secretions. Saliva contains an amylase, alpha amylase rather, that is, which can hydrolyze starch and glycogen. Alpha 1,4-glycosidic linkages can be hydrolyzed by the alpha amylase. But food stays in the mouth for a very short period. So contribution of salivary amylase in the digestion of carbohydrates is very small. Anyway, there is a now we say the one should properly chew the food before swallowing into the stomach. The more you chew the more it gets mixed with saliva, and a substantial digestion can take place. So for a good health, it is always advised to properly chew the food which we are taking. It is like they say, the solid food should be totally, it consistency should be like a liquid. Then only you should swallow into the stomach and further. So this is it is for a good health, one has to chew the food properly, adequately. Digestion of carbohydrates really begins in the small intestine. Stomach being high the acidic pH, there is carbohydrate digestion doesn't take place. So the next digestive secretion that can digest carbohydrate is the pancreatic juice. Pancreatic juice also contains the alpha amylase, and this acts an alkaline pH because the bicarbonate are being added, hydrolyzes the alpha 1,4-glycosidic bonds of starch and glycogen, converts starch and glycogen into maltose, isomaltose, and maltotriose. Maltotriose means 3 glucose residues are linked by 2 alpha 1,4-glycosidic linkage. That is a maltotriose. Isomaltose is the maltose where the 2 glucose residues are linked by alpha 1,6-glycosidic bond. Okay. And maltose is of course 2 glucose residues linked by alpha 1,4 linkage. amylase doesn't act on terminal alpha-1,4 glycosidic bonds and the alpha-1,6 glycosidic bonds present at the branch points. That's why we get a isomaltose. It cannot act on alpha-1,6 glycosidic bond. Intestinal secretion contains various disaccharides. The brush border of intestinal epithelium, they contain disaccharidases. These are enzymes, hydrolytic enzymes. So the name is according to the substrate. So maltase, isomaltase sucrase, it is a bifunctional enzyme, and lactase, which acts on lactose. Maltase acts on maltose, isomaltose and sucrase, they act on bifunctional enzyme and it acts on isomaltose and sucrose. Maltose hydrolysis leads to maltase hydrolyzes maltose and maltotriose into glucose. Isomaltose, as I was telling, it is alpha-1,6 glucosidase, which is different from alpha amylase. Isomaltase is alpha-1,6 glucosidase. So it hydrolyzes isomaltase into 2 glucose moieties. Sucrase or invertase, this is hydrolysis of sucrose into glucose and fructose. Lactase hydrolyzes lactose into glucose and galactose. It is a beta galactosidase. In the lactose, it is the galactose in the beta form linked to glucose. So it is a beta 1,4 linkage in lactose. So the enzyme lactase is, other name is beta galactosidase. It is lactose is a galactoside, not a glucoside. Lactose is both contains galactose and glucose, but the anomeric carbon of galactose is linked to 4th carbon of glucose. So it is a galactoside, and galactose remains in the beta form. So lactose structure, if you see, galactose in beta form, the anomeric carbon is linked to the 4th carbon of glucose. But glucose, anomeric carbon of glucose is free. So it is a beta galactosidase, or other name is lactase. It hydrolyzes lactose into glucose and galactose. Now, after these disaccharides, all are converted to the monosaccharides, and they can be absorbed through the intestine. So we'll see the absorption part. So this is the digestion part, and if some enzymes are deficient, then they can lead to certain pathological conditions, like hereditary lactase deficiency in children can cause lactose intolerance. Sometimes it is a bit rare, but it does occur. Babies, they cannot tolerate lactose, mother's milk, or any lactose-based product, and they will develop diarrhea, bloating, dehydration, etc. The mechanism is, in the small intestine, lactose should be metabolized to galactose and glucose by lactase. If lactase deficiency is there is accumulation of lactose will be there. It cannot be absorbed directly. So lactose will be acted upon by bacteria present in the intestine. So various metabolites, 2-carbon metabolites like acetate, or 3-carbon metabolites like lactate, or other hydrogen gas and carbon dioxide will be produced. So there is bloating in the intestine. So in the lower intestine, in the colon area. Then there will be diarrhea also, because this osmotic substance, they will absorb water, and watery diarrhea will be there. Dehydration, diarrhea may lead to dehydration. So hereditary lactase deficiency in newborn or infant can lead to all these lactose intolerance features, and they have to be withdrawn from mother's milk or any formula containing lactose. So other like soya or milk and other milks they have to be substituted, so their symptoms will improve. Then another is acquired lactase deficiency can occur in adults. Most of the adults they cannot tolerate lactose after certain age. As they grow older, our enzyme activity is reduced. It is not a mutation or something. It is only the activity lactose in the brush border of intestine decreases. So after certain years of age. So as you grow old, many people they cannot tolerate lactose, and they can also develop bloating, diarrhea, and other symptoms. And now another that isomaltase and sucrase enzyme, that is also sometimes hereditary deficiency in children of mostly seen in the Eskimos. So in that they will develop the similar symptoms like lactose intolerance after feeding with sugar, after taking sugar. So in that is not written here, deficiency of isomaltase sucrase enzyme can lead to similar symptoms, the bloating, diarrhea, dehydration, and other things after ingestion of sugar. And usually it is seen in the children of Eskimos. It is quite prevalent in Eskimos. Okay. So thus the major products of digestion of carbohydrates are glucose, fructose, and galactose. Small amounts of mannose, ribose, xylose, etc. may also be present in food. Cellulose cannot be digested in human beings, but its presence in food is important. Cellulose stimulates peristalsis. We'll see dietary role of dietary fibers. Stimulates peristalsis and helps in bowel movement. So that is one of the very important function. Proper bowel movement has to be there. So cellulose helps contribute the dietary fiber and helps in peristalsis and bowel movement. Now coming to absorption of monosaccharides, because only monosaccharides are being absorbed in the small intestine. This purpose of absorption is to transfer monosaccharides from the intestine into the circulation through mucosal cells. Monosaccharides are absorbed from the lumen of intestine into the mucosal cells first. There are two mechanisms for absorption of monosaccharides from the intestine. One is active transport, another is facilitated diffusion. Active transport occurs against concentration gradient. Energy is spent to move the compound from lower to higher concentration. A carrier protein is required for active transport. The process is saturable, whereas diffusion and they are not saturable, whereas this process. Glucose is absorbed actively from intestinal lumen in the mucosal cells by SGLT, sodium glut or sodium glucose transporter 1. There is similar transporter 2 in the renal tubules, which also does the same function. Sodium glucose transporter 2 present in the kidney renal tubules. SGLT1 or sodium glut present in the cell membrane has two binding sites, one for sodium and the other for glucose. Sodium and glucose present in the lumen bind to SGLT1, concentration of sodium in the lumen of the intestine is much higher than the mucosal cells. So sodium is transported into the mucosal cells down its concentration gradient. At the same time, glucose is transported into the mucosal cells against its concentration gradient. A rise in sodium concentration in the intracellular fluid is physiologically intolerable. The intracellular sodium concentration has to be kept low. For this, sodium is actively pumped out of mucosal cells into the capillaries. And pumping of sodium occurs against its concentration gradient. For every 3 sodium ions pumped out, 2 potassium ions move into the cell. The system that exchanges sodium and potassium ion is also known as sodium pump or sodium potassium pump. Ejection of sodium against this concentration gradient requires energy. Energy is provided by hydrolysis of ATP into ADP and PI. The hydrolysis is catalyzed by membrane-bound sodium-potassium exchanging ATPase, sodium-potassium ATPase enzyme. Here you can see the diagram. This is intestinal mucosal cell towards the lumen, and there is a transporter SGLT1, which has a 2 binding site, one for glucose, one for sodium. This has been magnified. And so sodium being less concentration inside the mucosal, sodium enters down the concentration gradient. Now glucose and to some extent galactose also can enter through the, although the concentration is, because sodium is being favorable, it can same protein, so the glucose or galactose can also enter into the cell. Now the sodium can rise after some times. It has to be maintained at low concentration. That is done by the sodium potassium ATPase pump, sodium potassium ATPase pump, where ATP is utilized for energy, ADP PI. 3 sodium are pushed out, 2 potassium are pushed in. So this is the sodium potassium ATPase. There is another protein which helps in the mostly fructose. It is facilitated. We will see what is the facilitated protein, GLUT4, that is mostly for fructose. Also some amount of glucose galactose can occur. And the other side of the, towards the capillary side, there is another GLUT2 is there. I'll come to what are the GLUTs. These are facilitated transfusion which requires the proteins, the protein transporters. This glucose transporter proteins are called glucose transporter. GLUT2 is the other side which helps in the diffusion of glucose, fructose, galactose to the capillaries. Okay, so like glucose, galactose is also absorbed similarly by SGLT1. Active transport is disrupted by OABANE, which is inhibited sodium pump. OABANE is a glycoside which can inhibit the sodium potassium ATPase. Belongs to digitalis family. So it inhibits the sodium pump. Active transport is also disrupted by fluoride, which displaces sodium from the carrier. Fluoride is a substance which can cause renal glycosuria. It can inhibit the SGLT2, thereby it competes with sodium and inactivates the sodium potassium pump, and so it can prevents the absorption of glucose also. So glucose will be excreted from in the urine. Anyway, so these were the this was the active transport. Now your facilitate diffusion. The other part is second is the facilitated diffusion, which requires some protein and membrane protein occurs down, and here it is always occurs down the concentration gradient. A carrier protein is required, no energy is required. Like active transport requires always energy, whereas facilitated diffusion doesn't require any. After a meal, fructose concentration in the lumen of the intestine increases. The concentration becomes higher than in the mucosal cells. Fructose is absorbed into the mucosal cells by facilitated diffusion. The carrier involved in fructose transport is GLUT5. There are many glucose transporters. Commonly, we 1 to 5. There are 12 or much more number of glucose transporters have been isolated and identified. But mostly we talk about the 5 types, 5 or 6, 7 types. So GLUT5 is present in the intestinal and the testis also, where it helps the transport fructose. It can also transport glucose galactose to some extent, but it is basically fructose. GLUT5 present in the intestine and in the testis also. Testis, we know the sperms, they are the main fuel. They take energy from fructose, not from glucose. GLUT5 is present on the luminal side of the mucosal cells of the intestine. Another transporter, GLUT2, is present on the contraluminal side towards the capillary side. GLUT2. GLUT2 transports glucose, galactose, fructose by facilitated diffusion. Concentration of glucose, galactose, and fructose in the mucosal cells increases after their absorption, and they are transported into the capillaries by GLUT2 down their concentration gradient. Once they are inside the mucosal cell, their concentration increases, so they can easily pass on to the capillaries through the GLUT2 protein or transporter down their concentration gradient. So again, the same thing has been shown here. GLUT1, this is the lumen side of the intestine, sodium being absorbed along the concentration gradient, so simultaneously glucose and galactose also can enter into the your mucosal cells. Now sodium intracellular has to be maintained low, so now sodium potassium ATPase helps in the ejection of 3 sodium and entry of 2 potassium. Energy is used, ADP-PI, and the GLUT5 also in the luminal side. Fructose is entering into the intestine mucosal cells, and this fructose, glucose, galactose, all of them are being transported to the capillary side. This is the capillary side through the GLUT2. This is a carrier protein for carbohydrate, glucose, fructose, and galactose. So monosaccharides released from internal mucosal into capillaries enter portal veins. Now this capillaries, they enter the portal veins. Their first destination is liver. All pathways of carbohydrate metabolism are present in the liver. So liver is after food, almost all the carbohydrates, they are being absorbed. So intestine and the portal vein, and they are delivered to liver. Liver release glucose into the system. From liver, it doesn't go directly to systemic circulation. From liver, it will be released to. If it is excess, liver will try to store it in the form of glycogen. So there is no need to release all the glucose into the systemic circulation. It will, otherwise it will be very high level of blood glucose after food. All the tissues take up glucose from circulation. So once liver releases the required amount of glucose into the circulation, the tissues take up glucose from the circulation. For uptake of glucose, tissue require glucose transporters. Different tissues possess different glucose transporters having different features. So we'll see, we are talking of the glucose transporter. These are all professor like GLUT 1, 2, 3, 4, 5 are there, and SGLT is there. So SGLT, the protein is active transport of glucose. So luminal side of enterocytes and renal tubular cells. Glucose, galactose, energy dependence, yes. Insulin dependence, no. GLUT 1 is present in erythrocytes. It has got a atherous brain and other, which dependent mostly on glucose. So specificity is for glucose, and energy dependence, no, and insulin dependence, no. It doesn't. GLUT2 is also present in the, we have seen control luminal side of the enterocytes. It is present in the liver and renal tubular cells. Also, it is present in the pancreas also. GLUT3 is present in brain and specific glucose. And GLUT4, most important here is GLUT4, adipocytes, muscles, and myocardium. Adipocytes, fat tissue in the storage of lipids, adipose tissue, muscles, skeletal muscles, and myocardium, heart muscles. All these, the glucose transport is occurring through GLUT4. And it is one of the important feature is it is insulin dependent. So insulin, if insulin is not there, so this GLUT4 are sequestrated inside the cell. In the presence of insulin, they will come to the membrane and they can help in transport of glucose into the adipocytes, into the muscles or myocardium. So in diabetes mellitus, the common abnormality we have seen in your clinical side, you will see the most common condition, diabetes mellitus, where insulin is actually or relatively deficient. In that condition, muscles and adipocytes, they cannot take glucose, so glucose will not be taken from the circulation. So there will be hyperglycemia. Common feature of diabetes is hyperglycemia. Blood sugar is very high. Why blood sugar is very high? Because from the circulation, the most maximum consumption should be muscles, skeletal muscles. They are very active. The energy, they require energy, and they take up continuously from the, but insulin deficiency, but it requires presence of insulin. And when diabetes, there is insulin deficiency is there, it cannot take up your glucose from the circulation. It will use alternate form of energy like free fatty acids, which will convert to beta acetyl-CoA and TCA cycle and all the things. So glucose will be high. So GLUT5 we have seen luminal side of enterocytes and liver, which is mostly from fructose and other sugar also. So these are glucose transporters present in the various tissues of the body, and they help in the active or facilitated diffusion of the glucose across the membrane. Now, this was how the glucose from the food are digested in the intestine, and they are absorbed into the circulation in the portal vein, rather capillaries and portal vein, and finally they are they pass to the liver, and liver again it will, depending upon its it will try to regulate the glucose. All of them will not be directly delivered to systemic circulation. When in excess, it will be stored in the glycogen form in the liver. So let us see what are the various fate of glucose or pathway involved in the metabolism of glucose. Carbohydrate metabolism is basically metabolism of glucose. Or of course, other minor sugars are there, fructose, galactose. They will also enter into the glucose metabolic pathways. Finally, glucose is the substance which provide energy to almost all tissues. So in the principal energy producing substance in our body is the glucose. That's why say blood glucose level has to be maintained, because glucose is the main energy producing, and many tissues are solely dependent on glucose. There are other energy producing substances could be there, like free fatty acids, ketone bodies, lactate, and other things are there, could be there. But glucose is the most important. Like RBC is totally dependent on glucose. RBC cannot use any other fuel. Brain is also mostly dependent on glucose for energy purpose. It is only, they say, extreme starvation, later stage of starvation, brain can use about 20% of ketone bodies. Again, majority are from glucose only. So, what are in the intermediate metabolism of glucose, the most common monosaccharides or more common carbohydrate in our body? A more important carbohydrate in the body is glucose, and it can, what are the various metabolic pathways involved? Anabolic pathway, one can see, like it can go for glycogenesis. Whenever blood, as glucose is being absorbed into the portal vein, goes to liver, excess of glucose is there, so it will go for glycogenesis. Glycogenesis is nothing but synthesis of glycogen from glucose. Okay, gluconeogenesis. When glucose level comes down, this synthetic pathway takes place, because as I was telling, most like RBC or your brain, they are mostly dependent on blood glucose for their energy purpose. But when blood glucose, somehow you are not taking food for a long time or any other reason, then glucose or insulin is not there, cells are not able to utilize glucose. So the process of gluconeogenesis can take place. Process of neoglucogenesis takes place. There is anaerobic neoglucogenesis is nothing but synthesis of glucose from non-carbohydrate sources. Synthesis of glucose for non-carbohydrate sources is called gluconeogenesis. So what are the non-carbohydrate? It could be amino acids, it could be fat. From fat, it is not the commonly the glycerol and the odd chain fatty acids. We will discuss that one. That can contribute for gluconeogenesis. Lactate is the common end product that also can help in the synthesis of glucose. Lactose is continuously produced by the RBC, and severely exercising muscles. So what is their fate? They can come to the liver, and by gluconeogenesis, glucose can be free form. And if glucose is excess, it can go for lipogenesis also. It can help in the lipogenesis. Is of course it is a mostly metabolic pathway of lipid metabolism, but here also we'll see how TCA cycle is involved in lipogenesis, and usually occurs when glucose is in excess. Glucose in excess, we'll see how lipogenesis can also take place. Catabolic pathways of glucose metabolism or carbohydrate metabolism are glycolysis. As you know, glycolysis is the most common universal pathway from where we get energy. almost all tissues, they can derive energy from glycolysis. Then glycogenolysis also is there. Whenever blood glucose comes down, whenever blood glucose is less, glycogenolysis can take place. Glycogenolysis, that means breakdown of glycogen into glucose. Glycogenesis is synthesis of glucose from synthesis of glycogen from glucose. Glycogenesis, the reverse pathway, not exactly the reverse pathway, the opposite pathway is glycogenolysis. Whenever blood glucose is less, glycogen has to be broken down to glucose to maintain blood glucose level. Glycogen is nothing but a reservoir of sugar. So sugar comes down, the glycogen will be broken down. Oxidation, it can also go to HMP shunt pathway. HMP shunt pathway, the glucose and the carbon metabolism, it is two important functions. One is your ribose phosphate. Ribose is very important for our body, and we need not take ribose in our food, but body can synthesize ribose for ribonucleic acid or deoxyribonucleic acid, DNA, RNA synthesis. Ribose is important. So from glucose, we can HMP pathway, shunt pathway, we can synthesize ribose. And another important is NADPH. NADPH is nothing but the reducing equivalent, which helps in the reductive biosynthesis of fatty acids, cholesterol, etc. So HMP shunt provides two important ribose and NADPH. We will discuss in detail subsequently. Also, uronic acid pathway. You have seen many heteropolysaccharides, they contain uronic acid, ioduronic acid, galacturonic acid, and other uronic acids. So they are also derived from glucose by uronic acid pathway. Many heteropolysaccharides, proteoglycans, and they contain uronic acid. So that can be obtained from glucose. So these are the, then the one thing is oxidation of pyruvate to acetyl-CoA. This is a link reaction between glycolysis and TCA cycle. TCA cycle is the final common pathway. Is the final common pathway where all the your proteins, carbohydrate, fats, they can be metabolized completely to give carbon dioxide and water. Of course, it is linked to respiratory chain. We have completed respiratory chain in the mitochondrial respiratory chain. So the end product of glycolysis will be pyruvate, mostly in aerobic condition, and that is converted to acetyl-CoA, which will enter TCA cycle. TCA cycle is not exactly anabolic or a catabolic, it is amphibolic pathway. Amphibolic pathway means it has role both in the anabolic pathways and catabolic pathways. For complete oxidation of glucose, glycolysis, at the end of glycolysis is partially oxidized, and it has to enter into TCA cycle for complete oxidation to get more amount of energy. That will be your catabolic pathway. Or still free fatty acids mobilized from the adipose tissue, undergoes beta oxidation in the mitochondria to acetyl-CoA. Acetyl-CoA again can enter into TCA cycle for complete oxidation to give high amount of energy. So TCA cycle is the final common pathway for carbohydrate, protein, and lipids for complete oxidation. Also, TCA cycle are involved in many synthetic pathways because like non-essential amino acids can be synthesized from TCA cycle intermediates. Non-essential amino acids can be obtained from TCA cycle intermediates. Alpha-ketoglutarate can form glutamate, or oxaloacetate can form aspartate. So you have seen in the heme synthesis also, succinyl-CoA is required. The first step of succinyl glycine for heme synthesis. So that is coming, succinylCoA is a component of TCA cycle. So TCA cycle intermediates are also involved in many synthetic processes. So that's why it is called amphibitic pathway. So we'll see so with this introduction and digestion and absorption of the various metabolic pathways of carbohydrate metabolism, we can straight go to glycolysis. Glycolysis is oxidation of glucose to pyruvate or lactate with the production of ATP by the embedded layer of pathway. You must be knowing all these in your 10th, 12th classes. This is the basic pathway, glycolysis pathway. So it oxidates glucose or glycogen to pyruvate or lactate. Two types: aerobic glycolysis or anaerobic glycolysis. Depending upon this, it can be either pyruvate or lactate. Aerobic glycolysis, breakdown of glucose in the cell with mitochondria in adequate supply of oxygen, is called aerobic glycolysis. Breakdown of glucose in the presence of adequate oxygen and intact respiratory chain, called aerobic glycolysis. Only oxygen presence will not ensure, it has to have a respiratory chain also, because oxygen, you have seen recently only biological oxidation, how the terminal acceptor of the electrons is the oxygen to form water. So presence of oxygen which is utilized by the intact respiratory chain, then the reducing equivalents are finally converted to water. So complete oxidation in the presence of oxygen and with an intact respiratory chain, it is called aerobic glycolysis. And end product will be pyruvate. And this pyruvate will go to, because intact respiratory chain is there, it can form acetyl-CoA. Acetyl-CoA will enter TCA cycle. We'll see TCA cycle will produce NADH or FADH2, which finally donate the electron to respiratory chain, and the end water will be formed, which is tightly coupled with phosphorylation, ATP synthesis from ADP and PI. Anaerobic glycolysis, breakdown of glucose in absence of oxygen, called anaerobic glycolysis. And please remember, glycolysis is the only pathway which can occur in the absence of oxygen also. No other pathway will be possible in the absence of oxygen. It is only glycolysis. So it occurs in cells without mitochondria, like RBC. So RBC, although it is full of loaded with oxygen, the glycolysis occurring in RBC is always anaerobic. That's why I told it is not only oxygen, a intact respiratory chain should be there, and the respiratory chain is present in the inner mitochondrial membrane. So if RBC doesn't contain mitochondria, there is no respiratory chain. So whatsoever amount of oxygen is there, that is not utilized. So it is an anaerobic condition. RBC, the cytosol of RBC contains the glycolytic enzymes, and it is always anaerobic glycolysis inside the RBC. So here the same pathway will be same, only the end product will be lactate in place of pyruvate. Why lactate? Because here you can see the till pyruvate it is same. If oxygen is adequate there, it can enter into the TCA cycle, oxygen and intact respiratory chain. But if it is not there, like in the RBC, intact respiratory chain is not there, so there is no TCA, no mitochondria, so no TCA cycle, no respiratory chain. It is So it has to go to other side. That means it will form lactate. Why lactate? Because in one of the step glycolytic pathway, it requires NAD, which gets converted to NADH. If normally oxygen and interrespiratory chain is there, the NADH crosses the mitochondrial membrane and gets reconverted back to NAD. During the process, what would be formed? But if mitochondria is not there or oxygen is not available, so this glycolysis has to continue. NAD can be regenerated without the help of respiratory chain, because there is no oxygen or no respiratory chain. So NAD, which are converted to NADH, they are again, NADH can convert pyruvate to lactate. In the process, it generates NAD. So this NAD can again, the triphosphate can form with multiple step pyruvate can be formed. So this can continue. Glycolysis process can continue with the production of lactate, even if there is no respiratory chain or mitochondria is present. Please remember these coenzymes, enzymes and coenzymes are present in very small amount in the cell. They are because enzymes are not reused, they are reused, it's not consumed. Similarly, these coenzymes are also in small amount, which are usually vitamins, derivative of vitamins, B complex group of vitamins. So vitamins are required in the diet in a small amount because they form coenzymes, and coenzymes and enzymes are present in the body very small amount. And this, if oxygen is not there, all the small amount of NAD will be converted to NADH. So further, if till trace NADs are not available, this pathway cannot take place. So NADH has to, in the presence of oxygen, there is no problem because respiratory chain is there. So NADH can cross the mitochondrial membrane from the cytosol, and it can donate the electron to respiratory chain and gets come back to NAD. If you have seen that in the respiratory chain, the first complex receives the NADH and the electron from NADH and converts back to NAD in the respiratory chain. Whereas in the RBC, there is no respiratory chain, so no mitochondria, so the NAD is regenerated by converting pyruvate to lactate, so that glycolysis will continue. Okay. This end product is lactate, and anaerobic glycolysis occurs in RBC, brain. Some amount of anaerobic glycolysis also occurs in, it is totally 100% in the RBC, some amount in the brain, retina, skeletal muscles, GI tract, mucosa, renal medulla, and skin. So in these tissues also some amount of anaerobic glycolysis takes place. So I will quickly pass through the steps of glycolysis. Glucose is converted. So once the glucose is entered into the cell, from the circulation, glucose will be taken up by the various tissues, depending on presence through the various glucose transporters. 1, 2, 3, 4, 5, but 5 is mostly fructose. So glucose will enter into the cell. So once inside the cell, it is immediately phosphorylated by hexokinase. Yes, it is hexokinase. It is not hexokinase. It is hexokinase only. It is a single word, hexokinase. So glucose is converted to glucose 6-phosphate. Hexokinase means kinases are enzymes which phosphorylate the substrate, and the product is a phosphorylated substance. And 6 kinase means glucose has 6 carbon, and 6th carbon contains the phosphate group. So glucose 6 phosphate, where ATP is used and ADP is produced. So 1 phosphate is added to the glucose 6 phosphate. This glucose 6 phosphate is advantage is it is being charged now. Once glucose has entered into the cell, it cannot diffuse back into the, because it is a charged molecule. It will remain inside the cell, and it can further undergo various metabolic pathways. We are talking about glycolysis now. So glucose 6 phosphate, there is another enzyme. Hexokinase is a constitutive enzyme. Constitutive enzyme means it is expressed in all tissues in a minimum amount, has to be housekeeping gene. This hexokinase gene is a housekeeping gene. That means housekeeping means in a house always the basic things are there, like salt, sugar, rice, wheat. Inducible enzyme, in contrast, inducible enzyme is as and when required. So housekeeping and inducible enzymes will be in inducible things is whenever some guests come, VIP guests, we bring to home some sweets or different items which are normally not present in our house, but because of somebody is coming, we will try to bring some good quality of sweets or some other substances which will be normally we don't keep always. Similarly, hexokinase is a constitutive gene, and a continuously small amount of produce amount is always there in most of the tissues, all the tissues. And there is, in contrast, there is another enzyme, inducible enzyme, is called glucokinase. Glucokinase is isomer enzyme of hexokinase, which is present in your liver mostly, and beta cell of pancreas and other sites also. So glucokinase is another isoform of which is liver contains hexokinase also and glucokinase. The difference between hexokinase and glucokinase, so hexokinase is a first enzyme of glycolysis present in almost all tissues. Glucokinase is present in liver and islet cell of pancreas. What are the differences? Affinity for glucose is low for glucokinase and high for hexokinase. That means Km value is low, here Km value is high. Specificity of phosphorylated or only glucose, specificity is glucokinase can phosphorylate only glucose, whereas hexokinase can phosphorylate any other hexoses. Location, as I tell, liver cell and pancreatic cells. Hexokinase is all tissues. Action of insulin stimulates glucokinase, induces rather, whereas hexokinase is constitutively not affected by any. Action of glucose 6 phosphate. It is hexokinase is allosterically inhibited by the product inhibition. Hexokinase is inhibited by the product, whereas glucokinase is not affected. So net effect is remove glucose from blood following meal, maintains blood glucose concentration anytime. Hexokinase, hexokinase can even if low blood glucose is there, because high affinity, low Km or high affinity, so like RBC has to depend on glucose. So even if blood glucose is low, Hexokinase can phosphorylate the glucose to glucose 6-phosphate, and glycolysis can continue in the low blood glucose constant also. Whereas liver mostly exposed to high blood glucose after meal, so it is not inhibited by its product. So, and it can deal with a large amount of glucose at a time, and all of them get phosphorylated, and they can undergo different pathways in the liver. So it can lead to take care the high influx of glucose after meal liver with the help of glucokinase. Now next step is this is the one irreversible step. Then this don't go truly, it is just a, yes, the reversible irreversible signs are not here clearly mentioned. So the steps are, as you know, phosphohexoisomerase, glucose is converted to fructose, then phosphofructokinase, fructose 1,6-bisphosphate will be formed. What is the difference between bisphosphate and diphosphate? Diphosphate is like ADP, adenosine diphosphate. Both the phosphates are consecutively attached. That means one phosphate, another phosphate is added to the first phosphate, second phosphate is added to the first phosphate. Whereas bisphosphate means two phosphates moieties attached to two different carbon, 1 and 6. So first fructose, first carbon, and 6th carbon, they are attached to phosphate group. So 1,6-bisphosphate, which is being acted upon by aldolase A. Gets it is a which cleaves the molecule 6 to 2,3 carbon moieties, dihydroxyl phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate dehydrogenase enzyme then converts it to 1,3-bisphosphoglycerate. This is a high energy compound. Here initially we are activating the glucose by spending some energy. ATP, 2 ATPs are to activate the molecule. But then subsequently we are getting ATP energy. We are getting from 1,3-bisphosphoglycerate, 3-phosphoglycerate is being produced. This is where two molecules of ATP gets called 2 ATPs. This is called substrate level phosphorylation. We have seen ATPs are produced mostly in the respiratory chain because oxidation is linked, tightly coupled to phosphorylation in the respiratory chain. Proton pumps and proton are pumped out across the inner mitral membrane, and finally sodium potassium ATPase that forms the ATP. So majority of the ATPs are produced by the respiratory chain, but in the glycolytic pathway energy we are getting at the level of substrate. So it is called substrate level phosphorylation without the use of respiratory chain. 2 ATP, 2 ATP, then this forms 3-phosphoglycerate. 3-phosphoglycerate is mutase enzyme. It converts to 2-phosphoglycerate. Then by enolase enzyme, it gets converted to phosphoenol pyruvate, and which finally, with the help of pyruvate kinase, forms pyruvate. Here is also another example of pyruvate kinase, example of substrate level phosphorylation. Then fluoride inhibits enolase. Fluoride will be inhibited by fluoride will inhibit the enolase enzyme. So what is used? These fluoride tubes are used to collect blood for estimation of sugar, blood sugar, because blood sugar, that is called sugar vial. We have seen with different vials are different color coded vials are there to collect blood. If you see any collection center, there are they keep, or the person who collects pink color, gray color, or different capped containers are there, tubes are there. So fluoride for the gray color background, that is for specifically for estimation of blood glucose. Why blood glucose needs a fluoride extra? It needs usually plasma glucose is measured by adding anti-ADTA and fluoride. Why? Where ADTA is simple anticoagulant, but why fluoride? Because if you don't estimate blood glucose immediately and keep for few hours before estimation, then the RBCs in the blood, they will use the plasma glucose, because RBC uses glucose for its glycolytic pathway, anaerobic glycolytic pathway. So if you keep 1 or 2 hours after collection of blood glucose, plasma glucose level will come down. So to prevent the RBC's utilization of plasma glucose, we add fluoride, which inhibits enolase. So once enolase is inhibited, so all the pathway will be, the glycolytic pathway will be inhibited, and RBC cannot utilize glucose. So usually it happens, we do collect blood in the early morning, or some are in the collection. So large hospitals, they collect together and they are transported to the actual site of estimate autoanalyzers, where machines are there. So during that process, collection process and actual estimation of blood glucose by the machine, there is a gap of 2-3 hours. So if you don't add fluoride, blood glucose will come down and it will give a false low value. So we always estimate blood glucose we collect the blood glucose for estimation by a fluoride vial or gray vial called fluoride is added. So let us stop here today because of this some technical problem. We could not start in time, so I'm also not able to finish my scheduled glycolytic pathway TCA cycle both. So thank you. Any question you can email, or I don't know, you can ask, or in the tutorial class we can discuss any problem. And so in the next class we'll discuss the more details of glycolysis, steps of glycolysis and their regulation, then subsequently how pyruvate is converted to acetyl-CoA and enters TCA cycle, and how complete oxidation of glucose in the aerobic pathway get much more energy than the anaerobic pathway. All these we will discuss in the next class. Thank you. कोई है पीजी को बुलाओ, पीजी रूम में कोई है, डॉक्टर राम या कोई नहीं है, बुलाओ, इसको बंद करना पड़ेगा। बुलाओ।